Hi, I'm Yiren. I'm a senior research specialist at ASU Central Arizona Phoenix Long-Term Ecological Research. I'll be your guide for the Tempe Town Lake Water Quality Project Field Lab Research Experience. As a part of the CAP LTER volunteer opportunity, you will assist with water sample collection and maintaining calibrating a high-precision data sound deployed in Tempe Town Lake, a dynamic man-made reservoir in the Salt River bed. This experience offers a unique chance to enhance your research resume and to work closely with CAP LTR field and lab scientists in a real-world research setting. I'm very excited to connect with you and look forward to help you with research questions. If you have any questions, please email me at yren45@asu.edu. Before we dive into the details, let's go over the required trainings. To participate, you must complete 3 ASU training courses: laboratory safety training, fire safety training, and field safety training. These courses are available online through Canvas. Details regarding where to take the trainings can be found on the project description page. Please make sure to complete these trainings and email me a proof of completion at least 2 days before the scheduled date. Let's take a closer look at the Tempe Town Lake project. Tempe Town Lake is a 224-acre man-made reservoir that transforms a section of typically dry Salt Riverbed into a thriving community and ecological resource in the heart of Tempe. The lake's design includes a hydrologically operated steel gates that allows water to flow through during river activity, balancing flood control and urban use. Tempe Town Lake is an ideal model for many artificial lakes constructed in area-land cities because it reflects its ecology from management decisions such as draining and dramatic shifts in hydrology and chemistry when the lake is transformed to a flowing river and back into a lake during and after flood will provide opportunities to study this dynamic system. CAP LTR has been monitoring the lake's water quality since 2005. We measured key parameters such as temperature, pH, conductivity, dissolved oxygen, dissolved organic carbon, and total dissolved nitrogen. Back then, these data were collected using handheld meters and laboratory analyses. And now, since 2018, we transitioned to using an in-situ data sound, which is capable of collecting high-resolution data every 30 minutes. This allows us to transition from manual sampling to continuous monitoring. The activities we'll be doing involves field work at Tempe Town Lake, where you will help collect water samples and retreat and then deploy the data sound. And we'll have lab activities focused on calibration and maintenance of the data sound. Hopefully, these will help you gain hands-on experience while taking a peek into the process of ecological research to discover the future interests in your scientific research journey. Before heading to the lake, let's review some field safety basics. Tempe Town Lake has specific characteristics to be aware of, such as slippery and swinging dock during a stormy day, and of course, the Arizona heat. Always wear comfortable shoes. Bring some water even though we're not gonna stay outside for too long. When preparing to visit Tempe Town Lake, we will go through the field safety document to refresh our knowledge about the potential safety hazards at the site. It's important to double-check packed essential items such as life jackets, gloves, and other equipment for water sampling and retrieving the sound. Please pay close attention to your emails in those couple days before the scheduled date, because we will reschedule if it is really windy or if the dock is wet. At the lake, we'll record anything abnormal we see at the site, retrieve the sound, and do initial cleaning of the sound with a brush and bucket at the dock. Then we will take 3 water samples and filter them on site for dissolved organic carbon analysis. Then we will bring the sum back to the lab and do calibration and maintenance. After that, we will return the sound back to the dock. After coming back to the lab, we'll first clean the sound thoroughly using brushes and Scotch-Brite to make sure the sensor area is cleaned so the calibration won't be interfered. We'll then download last month's data, calibrate and check the accuracy and response factor of all sensors, including pH, conductivity, turbidity, depth, dissolved oxygen, CDON, and chlorophyll sensors with a variety of calibration standards. After calibration, we'll replace the battery, set up logging files, and reassemble the sound for deployment. Let's get into the details of each parameter measured by the data sound, and how each water probe functions. First, we have the pH probe. pH is a measurement of water's acidity or alkalinity. Water with pH less than 7 is acidic, and water with pH greater than 7 is considered as alkaline, which is basic. The pH of water is determined by dissolved pH-active salts. For example, dissolving sodium hydroxide in pure water will increase the pH because the hydroxide ion is a pH-active base. And dissolving hydrogen sulfide in pure water will decrease the pH because the hydrogen ion is a pH-active acid. But dissolving sodium chloride table salt in water will not change the pH because neither sodium nor chloride is a pH-active ion. The pH value of water can be used to determine the form of dissolved pH-active salts. For example, when an ammonia-based fertilizer flows into the river through runoff, if the pH of river is above 9, most of the ammonia species will be present in the form of ammonia, NH3, which is toxic to biota. If the river pH is below 9, the ammonia species present will be mostly in the form of ammonium, NH4+, which is a nutrient for biota. pH value can also be used to determine the solubility of materials in water. A good example can be that iron is more soluble in low pH, so it is more available for uptake by fish and other biota. At a higher pH, the metals are less soluble, so they're more likely to bond into sediments than available for biota. The changes in the long-term pH trend can tell us the signal of the need for more detailed chemical study of the water and also its contamination sources. pH is measured with a pH sensor and a reference electrode. The pH sensor is a small glass bulb filled with an electrolyte of a known pH. If the pH of the sample, which is water, is different from that electrolyte, a voltage is created across the glass bulb. That voltage changes predictably with the pH of the sample water and is measured through the reference electrode. The reference electrode contains an electrolyte separated from the sample water not by a glass bulb, but by a material that is microporous, and in this case is Teflon. It allows the electrolyte to contact with water without being quickly diluted by water. Since the pH value at Tempe Town Lake is relatively stable, our pH probe will be calibrated with only a 2-point calibration with known buffers of pH 7 and pH 10. In other studies where the pH can be significantly more variable or requires a high accuracy, a 4-point or even 10-point calibration can be performed. Now let's move on to conductivity. Conductivity is the ability of water to conduct electricity. This conduction requires ions to present in the water to be charge carriers. Pure water contains almost no ions, so it is a very poor conductor of electricity. But as water picks up chemical salts, those salts add ions to the water when they dissolve, so as the ionic strength of water rises as well as the conductivity. So the saltier the water, the higher the conductivity. But the conductivity measurement cannot tell you the concentration of ions in water, because different ions have different contributions to conductivity. A large ion like acetate do not help conduct electricity well. A smaller, faster ion like H+ make a much higher per ion contribution to conductivity. So natural waters are usually have a mixture of ions, and the conductivity reading is only a relative measurement of ion concentration. Conductivity can be used to monitor the stability of a water body. An unchanged conductivity usually means there's no chemical or being added to or subtracted from the water. A spike in conductivity often indicate an increase in ion from a pollution source such as wastewater or agricultural runoff, and a dip in conductivity can indicate a reduction of chemical input or an increase in freshwater input. Conductivity reading can also be used to judge biological health. For example, oysters does not do well in freshwater, and freshwater fish do not do well in salty waters. The conductivity sensor in the data sound is a 4-electrode sensor operated by measuring the current required to maintain a fixed voltage between two pairs of electrodes separated by the water. The conductivity probe is calibrated with one standard, which is a solution with known concentration of potassium chloride. Now let's move on to turbidity. Turbidity is an indication of the amount of light that will pass through a column of water. Clear waters have low turbidity, and waters with high suspended solids have high turbidities. Turbidity is measured for several reasons. First, it is a gross indicator of the aesthetic value of a water body. A river with a low turbidity looks clear, and high turbidity looks muddy and discolored. Second, in a low turbidity water, the transmission of sunlight is maximized for photosynthetic productivity. So clear lakes will have higher biological productivity because minimum amount of sunlight is blocked by the suspended particles in water. Third, high turbidity reduces the ability of higher life forms, and low turbidity makes the smaller fish more subject to predation. In natural waters, turbidity is most often measured by an optical sensor. The amount of light scattered by suspended particles at 90 degrees to a beam of infrared light. In very clear waters, little light is scattered and the turbidity reading is low. But in turbid waters, much light is scattered by suspended particles and the turbidity reading is high. The calibration of our turbidity probe is a 2-point calibration with air and a known concentration of formazine standard. Now let's move on to dissolved oxygen. Liquid water is a loose matrix of water molecules. If water with oxygen come into contact with a source of oxygen, for example the atmosphere, the oxygen from the atmosphere will slowly move into the gaps in the water molecule matrix, and that oxygen is called dissolved oxygen. It's the same thing as dissolving table salt in water. Oxygen dissolving water usually come from the atmosphere or photosynthetic organisms such as algae in the water. Just like humans, fish and other aquatic animals must breathe oxygen to live, and they are adapted to breathe oxygen dissolved in the water instead of oxygen in the atmosphere. A low dissolved oxygen levels can cause fish kills, which are often triggered ironically by the decomposition of algae that have been adding oxygen to the water before death. Dissolved oxygen also influences basic water chemistry. Highly oxygenated water will favor the oxidized form of chemicals, and the poorly oxygenated water will favor the reduced form of chemicals. For example, oxygenated water favors sulfur in the form of sulfate, SO4, while deoxygenated water will favor sulfur in the form of hydrogen sulfide, H2S. Hydrogen sulfide is a poison, but sulfate is a much softer substance. The type of dissolved oxygen sensor we have on our data sound is an optical dissolved oxygen sensor. The blue light from the sensor causes the oxygen active compound to fluoresce. They absorb energy in the form of blue light and then emits energy as red light. So the emission of red light is reduced if oxygen molecules are present in the interface of the oxygen active compound. The more oxygen present, the smaller amount of red light will be produced. Now let's talk about chlorophyll. Chlorophyll measurements tells us the population and distribution of phytoplankton or algae. It provides a lot of information about an aquatic system's health and composition. Chlorophyll is the pigment that makes photosynthetic plants look green. The amount of chlorophyll present in water indicates the level of photosynthesis. And the more photosynthesis, the more primary food production. Without photosynthesis, there would be little to no aquatic life higher in the food chain. And that is why chlorophyll measurements are a good indication of the amount of solar energy that has been converted into sources of food for everything from zooplankton to fish and other aquatic species. But too much chlorophyll are often an indicator that the water body has become eutrophic. Eutrophic water looks unappealing, can produce taste and odor problems in drinking water treatments, and it also can cause death of fish when the photosynthetic organisms such as algae die and decay rapidly. The chlorophyll sensors in our data sound is a fluorometric sensor. Fluorometric sensors emits light at a certain wavelength, and you look for a very specific different wavelengths in return. If you shine a blue light at chlorophyll, the chlorophyll absorbs some of the blue light and then emits red light. More chlorophyll produce more red light. The magnitude of returned red light is relatable to the amount of chlorophyll present in the water sample. Okay, let's talk about CDOM. CDOM stands for Colored Dissolved Organic Matter. So dissolved organic matter, DOM, exists in many forms, from naturally occurring humic acids to byproducts from organisms. DOM is a highly abundant form of organic matter and represents a major reservoir of reactive carbon. It is also a dynamic substrate that can undergo reactions to become accessible to bacteria, plants, and even animals as an energy source. It can also photodegrade, which will produce volatile compounds that can have adverse effects on organisms and the environment. DOM typically contains chromophores that absorbs UV and visible light, and that is why we call it colored dissolved organic matter CDOM. And CDOM will also fluoresce after light absorption, so we also call CDOM FDOM. The detection of CDOM concentration in water system can help us explain events such as a sudden decrease in primary production or algae blooms or other changes in an environment. But CDOM is only a portion of DOM, as CDOM measurement cannot represent the whole dissolved organic matter pool in the water system. That is why we will take water samples for dissolved organic carbon measurements using the DOC analyzer. It will be very interesting to look into the data and compare CDOM data to the DOC data at Tempe Town Lake. The CDOM sensor on our data sound is a fluorometric sensor which works the same way as a chlorophyll sensor. It emits light at a certain wavelength and looks for very specific different wavelengths in return. So that kind of concludes the overview of this volunteer opportunity. We will get more into it throughout the day when we meet up. But please remember to complete the required trainings and send me a proof of them at least 2 days before the scheduled date. And please email me if you have any questions. Hope to see you soon.